to you about some of the work that I did now a couple of years ago during my PhD in Oxford. This is joint work with Ellen Byrne in Oxford and Panos Kevrakidis from Amherst, Massachusetts. And so today I want to talk to you about the connection, how we use mathematical modeling to try and understand the connection between hypoxia, cell reprogramming, so one of the hallmarks of cancer that we've seen yesterday and Thomas Organ introduced very well, in aiding the growth and the dynamics of the tumor. So as we have said many times in this meeting before, the tumor is, cancer is a very complex multi-scale ecosystem. And what we know is that when we zoom in, not only we have heterogeneity both in the microenvironment cells are exposed to, as well as heterogeneity in what the cells are doing themselves. So one of the key aspects of the tumor microenvironment is oxygen levels, so nutrients. The idea is that in tumors, unlike in normal tissues, because of the imbalance between cell growth and consumption, we have the formation of region of extremely or pathologically low oxygen level that we usually call hypoxia. And in contrast, we have also cells that are exposed to normal oxygen level, what we call physioxia. And so why people have been interested a lot in hypoxia is that this is a very stressful situation for cells. And so what cancer has done is that it's used all of several mechanisms that cells have from development to try and adapt and survive this very extreme situation. And so hypoxia actually connects at the cell level to several of the hallmarks of cancer that we have seen yesterday, like cell cycle dysregulation, VEGF production, and angiogenesis at downstream. People have connected this to EMT, but what I want to focus about today is particularly the connection between hypoxia and stemness. So this idea of differentiation, the differentiation of cells. And the idea is that this cell level connection between what cells reprogramming and the tumor microenvironment eventually we know affects the overall dynamics of the tumor as well as the response of patients to treatment. And so really our general goal has been to try and understand using mathematical modeling how hypoxia contributes to the tumor progression and also response to treatment. But since I have limited time today, I want to talk to you, focus especially on the first part of this project, which has been investigating the connection between oxygen level hypoxia and cancer development in the absence of treatment. And so I'll divide my talk into three sections. I'll give a little bit about of background about the biology. Then I'll move on to present our modeling framework and some of the key results and analysis. So about the biology and stemness. So this is associated, what people have found now 10 years ago is that similarly to normal tissues, also tumors have a hierarchical structure. So we can identify cancer stem cells. These usually are maybe polyclinic cells that are highly tumorigenic. So they're the one that basically are a pool of new cells and drive the growth of the tumor. And they're usually also highly resistant to treatment. Then cancer cells can differentiate into differentiated cells. These are usually forming the bulk of the tumor, and they're highly proliferative cells that, however, have final reproduction capacity. So eventually, they become terminally differentiated, and the only thing that they can do is die. You can maybe call this senescence. We don't really know. But so the idea is that what people had proposed initially was this cancer cell hypothesis in which, similarly to normal tissues, also cancer follow this development path. And so you have cancer stem cells that differentiate and eventually they become terminally differentiated. And this is a one-way project, a one-way process. But actually what they found is that cancer can hijack this mechanism, and actually when exposed to the proper signal, it can de-differentiate, one of the hallmarks of cancer that we're seeing yesterday, and so go back into this state of high tumorigenic infinite proliferation capacity. And it's really when they're exposed, cells are exposed to toxic environments such as hypoxia, this reprogramming can occur. And so our question has been like, given this general framework, so using a bottom-up approach, given this biological understanding, can we draw some conclusion on how this affects the emergent dynamics of the tumor? And to do that, we use what we call phenotypic structure model that probably several of you have already heard about. So it's a theoretical work. So we start with several simplification. We consider a slice of tissue that is oxygenated by a vessel. By assuming symmetries around x equals 0 and homogeneity in the y direction, we just simplify for the purpose of the initial analysis to a 1D case, and I'll present all of the results in non-dimensional variable for simplicity. But so we start abstracting to simplify and understand really how this mechanism and this biological C, sorry, is the oxygen level. So produced, by the way, is maximum at the vessel, fixed, and then it diffuses as it's consumed, usually we form gradients. That's a great question, sorry. And so this is going to be a function of space as well as time. And so at each location in space, we have a collection of cells. So we can describe this by the number of cells. But the key idea is that, as we have seen before, cells are not homogeneous, but they're structured by the level of stemness. And so really the way in which we describe this system is by counting the number of cells that have a certain phenotype. And we call this function the local phenotypic distribution. So at each location in space, for each value of this phenotype that goes from stem-like cells to fully differentiated cells, we count the number of cells that have the specific phenotype. And we want to describe how this function changes both in space as well as in time and as a function of S. So how we describe the time evolution of this function? Well, we assume that cells can move. And for simplicity here, we start with the simplest type of operator for movement, so cell diffusion in 1D. Then we have, of course, that, as we know, cancer cells can proliferate but also die. And so this is a key, usual sink and source term, where one of the key aspects is that we include cell competition, and we assume that all cells compete with each other independently of their phenotype. And so this is kind of standard kind of selection type of mechanism, which is mediated also by oxygen C. But then the key idea is that we want to describe how cells evolve along this phenotypic axis. And we have two key mechanisms. We assume that there is some random diffusion. This is because at the phenotypic level, there might be fluctuation or noise in how cells behave. And then the key aspect is that we have this advection term, and this captures the effect of the environment, so C, our oxygen level, on the behavior of the cells. So this is really where oxygen hypoxia-mediated cell reprogramming happens. And if we look at the form that we use for this function, we use probably the simplest form, where we basically interpolate continuously from differentiation in a high oxygen level. So as you see, the highly blue, and then the differentiation in the region of hypoxia. So below a certain threshold of oxygen, cells kind of revert differentiation. And then we couple, we need to couple the evolution of the local phenotypic distribution to the oxygen concentration in the tissue. And so again, here we assume that nutrients diffuse while they are consumed by the cell. And here we assume that the consumption is homogeneous. And we have one of the key parameters here, which is this gamma naught, that basically controls the consumption of oxygen by cells. And in some way, this controls the level of spatial heterogeneity in the tissue. And so what we obtain is a system.
Sorry, why is there a threshold?
We just assume that when basically the consumption is constant, and it just becomes zero below a certain threshold. So they consume oxygen at a constant level, and then when it's too low, they just stop consuming oxygen. This is used in previous work, by the way. It could be proportional to see. It needs to go to zero at some point, but yeah. So what we obtain is a system of coupled nonlinear and non-local PDEs for the evolution of the local phenotypic distribution and oxygen, where we have highly nonlinearities in the dependence of the oxygen on our advection function as well as proliferation. And then we have also competition when we introduce nonlinearity, and the non-locality comes from these integral terms and from the fact that we have an heterogeneous population. Okay. And of course, we need to couple this with a boundary condition, proper boundary condition and initial condition. So we can take some of the value of the parameters from the literature, as we usually do, and start running some simulation to understand what are the prediction of the model. And so here I'm showing you two different simulations. As you can see on the left-hand side, we have two tumors. They both initially grow, but then eventually the red line, the one on the left, kind of goes towards extinction, while the blue curve that represents the tumor on the right grows and eventually saturates to a carrying capacity. And when we look at these structures on the x-axis, we have axis space. So we see spatial heterogeneity in the distribution of the tissue, and we see that the type of cells that we observe, so the phenotypic composition is different in the highly oxygenated region compared to the poorly oxygenated region.
What's the difference between blue and red?
The initial tumor burden. So all of the parameters are exactly the same. The only thing we're changing is the initial condition. So we see a little bit more cells on the right-hand side compared to the left-hand side. So this is a clear sign of multi-stability. And so we investigate a little bit more what drives the multi-stability in the system by using the dynamical simulation. So now what I showed you, we parameterize the initial condition, so a kind of homogeneous population of cells by the initial tumor burden, so how many cells I see, as well as the initial phenotype, the phenotype of the population. And so we can do this phase diagram in which I have the mass on the vertical axis and the phenotype on the horizontal axis. And what we find is that there is a clear dependence interplay between the mass and the initial phenotype in deciding what the eventual long-term behavior of the system is. And particularly, we find that for any fixed S0, so if we fix the initial composition of the tumor, then there is a critical mass above which the tumor fully developed, and below that mass, the tumor goes to extinction. And what is key is that this critical mass depends on S0. So it depends on how stem-like the initial tumor is. So if we have a highly stem-like tumors, then this critical mass is very low. While if we start with fully differentiated, already exhausted cells, no matter how many you put there, they would eventually go extinct, even a very high mass. So there is a dependence both on tumor burden as well as phenotypic composition on the long-term behavior of the system. And this, if you think about it, like from the ecological perspective, was called an Ali effect. So the fact that we have this threshold mass to drive the formation of the tumor, but this Ali effect is mediated by the heterogeneity, so the phenotypic composition of the tumor. And so we want to really understand a little bit more where this mechanism is coming, where this bistability is coming from, and whether it's something robust in our system. Were we lucky? Is actually something intrinsic. We can, there are two key ingredients to have an Ali effect. First of all, we have to have stability of the trivial steady state, so the extinction in this case. We can study it analytically, and what we find is that there are two key parameters. There is one key parameter that decides the stability, that drives the stability of the trivial steady state, which is V+. I'll get back to it in a second. But then we see that this stability is independent from the consumption of oxygen, this gamma naught. And numerically, we can actually characterize the change in stability, the bifurcation that changes the stability of a trivial steady state, which is a transcritical bifurcation. And so just to remind you, V+, the key parameter that decides the stability of the trivial steady state, is basically developing the time scale at which differentiation occurs in high oxygen. And gamma naught instead was our consumption of oxygen. And then we have to look at the non-trivial steady state that needs to be stable as well to have this bistability switch. And we do that numerically using something called numerical bifurcation analysis. I'm happy to talk more about it, but this is a link to the paper where we describe it in more detail. So the key idea is that for different value of our consumption, gamma naught, I can compute basically steady states of my system. So if we do gamma naught to be large enough, what we find is that we have different regimes depending on the velocity of the differentiation. So if we have, first of all, we have that if V plus is sufficiently large, we have that extinction is possible. And that's what we have seen before. Then we have another branch of the solution, this mid branch between the yellow dot and the red dot, the saddle node bifurcation, which is basically an homogeneous tumors that is basically spatially homogeneous and is composed mainly by fully differentiated cells. So you would think about this as a non-invasive and avascular tumor. Then we have another branch, which is instead characterized very high tumor burden and which is spatially heterogeneous. And that's exactly what we observed in the simulations. And so we have that instead, when we have, and we see that basically the emergence of these two non-trivial solutions is from a saddle node bifurcation. But these two saddle node bifurcation eventually collapse into each other as a function of gamma naught. And what we find is that in the absence of oxygen heterogeneity, we just have two types of solution. And we have that either the system is, the trivial solution is stable, or we have a non-trivial solution, which is stable. So we do have a range of possible regime we can be in, depending on the two parameter gamma naught and V plus that determines the number of solution and the type of solution that we observe. But what we find is that by doing a co-dimension 2 bifurcation analysis, so by looking basically on how these two saddle nodes move in the parameter space, the largest area that you see is this light green. Unfortunately, it's difficult to see because of the light, but this is the large range of parameter values that we observe. And this is exactly where the Lee effect, of course. So we were not just lucky. It seems like for the large range of the biologically relevant time scale of the processes, we do observe this Lee effect. And so coming to the conclusion, I've showed you that the interplays between hypoxia and cancer cell reprogramming can give rise to very complex emergent dynamics in the tumor behavior. And especially we do observe that the long-term behavior of the tumor is not determined only by the initial tumor burden, so the mass of the tumor, but also its composition. And in work that I haven't had time to talk to you about today, we kind of use this model and introduce radiotherapy to see how this finding affects optimal strategies in terms of treatment. And what we find is that because of the interplay between the tumor burden and the composition of the tumor, treatment holidays can be sometimes effective. So giving more time in between doses can actually help us have a better outcome. And so looking at future work is what I would like to do during my time here at the field, is that trying to use similar techniques and similar approaches to study heterogeneity in another of the hallmarks of cancer we talked about yesterday, which is senescence, and trying to understand this pro-anti-tumor impact of senescent cells and see whether mathematics can tell us something about this paradox. And with that, I'd like to conclude. Thank my collaborators, my funding, and if you want to chat, my office is 4:30. I always like to have a coffee, and these are some references, and I'm happy to take any question or discuss throughout the week. Thank you.